Assalamu alaikum. So we'll talk about transcription today, and this is going to be a long lecture. So bear with me. Again, the main resource is this lecture, but you can refer to Cooper's textbook. There are 3 pages in the introductory chapters, and you can refer to chapter 8 for this lecture and the other topic, the next topic, which is regulation of transcription. So let's start. Let me start by defining what a gene is. Now you will find in textbooks and different resources different types or different definitions of what a gene is. Now this is my definition, so pay attention to that. Okay, what I've tried to do is I've tried to define what a gene is according to what we will cover in these lectures, what my understanding is, what I would like you to know, and what also exists in different resources. So a gene is the following. It is the entire DNA sequence. So we're talking about a specific region in the DNA, in our genome, or any other organism's genome. So it is a region in the DNA, in chromosomes. This region is necessary for the synthesis of a functional RNA. Now, this RNA molecule can be messenger RNA, ribosomal RNA, transfer RNA, long non-coding RNA, microRNA, or any other type of RNA molecules from this long list of RNA molecules. Now notice that here I'm saying that it is necessary for the synthesis of a functional RNA. So it can be a region that is transcribed, so producing an RNA molecule, or it can be a region that is necessary, that regulates transcription, that is necessary for transcription. So it can be something like a promoter, an enhancer, a silencer. It can be any one of these regulatory regions that are not transcribed, but they are necessary for transcription. Okay, and we will talk about all of these different regions during these lectures. So if you have the production of a messenger RNA, this messenger RNA can be produced to synthesize a polypeptide. So it's basically the entire DNA sequence that is necessary for the synthesis of a functional RNA, okay, or a polypeptide which is produced from messenger RNA. Now notice that here I did not say the production of a protein, because there is a difference between a protein and a polypeptide. A polypeptide is just a stretch of amino acids with no particular structure. A protein has a three-dimensional structure and it has a function. A polypeptide may not have a function. In addition, this polypeptide can be cleaved into smaller peptides, and these peptides can be functional. Okay? So, again, it's a DNA, it's a region in chromosomes that is necessary for the synthesis of a functional RNA. This RNA can be translated like messenger RNA, or it may not be translated like ribosomal RNA, tRNA, long non-coding microRNA, all of the other RNA molecules. Now this messenger RNA can be used to synthesize a polypeptide. This polypeptide can then have a three-dimensional structure, thus called a protein, or this polypeptide can be cleaved into smaller peptides, and these smaller peptides are functional. So the DNA sequence, again, it includes the coding region that makes proteins or other regulatory sequences like promoters, enhancers, and so on. Now, it also includes non-coding regions like introns, and we'll talk about introns as well. Now, there's another term for genes, and it is an older term, and this will help us understand transcription and translation in prokaryotes versus eukaryotes. And this term is known as a cistron. Now, a cistron is an alternative term of a gene. Now, pay attention to this. If this cistron encodes one polypeptide from one messenger RNA, it is called monocistronic. Again, so you have a region in the DNA. This region can produce one messenger RNA, and this one messenger RNA can produce one polypeptide, okay, not necessarily protein, then it is called monocistronic. Now, if this region encodes a messenger RNA molecule, and this messenger RNA can be used, mature messenger RNA can be used to produce different polypeptides from different regions, then we call this cistron we call it polycistronic. Okay, so monocistronic and polycistronic. Monocistronic, you have one messenger RNA producing one polypeptide, and polycistronic is basically one messenger RNA producing different polypeptides from different regions of this messenger RNA. Okay, so remember that. So let's talk about the general mechanism of transcription. And usually I like to start with prokaryotic systems, since again they're simpler and more understood. Let's define what transcription is. Transcription is basically the process of making RNA from DNA. That's it. Now, in transcription, we use one of the two strands of the DNA, and this strand is used as a template to synthesize this RNA molecule. Now remember, in DNA replication, both strands act as templates for the daughter strands, but in transcription we use one strand for any particular gene or cistron. So this is what I mean. Let's say that in this particular chromosome or DNA, there are three genes: gene 1, gene 2, and gene 3. Now, that's what we say. So we say there are three genes located on this site or region in the DNA or chromosome. Now, for gene 1, it's this strand that is used to synthesize the RNA molecule. Now, same thing with gene 3 or for gene 3. It's this strand that is used to synthesize the RNA for that gene. Now, but for gene 2, it is the other strand that is used to synthesize the RNA molecule. So both strands can be used as templates, okay? But for any particular gene, only one of the two strands is used as a template, okay? So what does determine which strand to be used as a template for that particular gene? It's this right here. This is known as the promoter, and we'll talk about promoters right now. These regions are the RNA polymerase binding sites. So once the RNA polymerase binds to the promoter, binds to this region, it guides the RNA synthesis in that direction. That is, in the direction of 5' to 3'. Again, we'll talk about that. Now, on the other hand, for gene 2, the promoter region is located right here on the other strand. So the RNA polymerase would bind to the promoter region, and again, it would synthesize RNA from the 5' to the 3' end. Okay. Same thing here. Now, notice that the template is read from the 3' to the 5' end, or in the direction of 3' to 5'. Okay? Now, So RNA is complementary to its DNA template. Remember that. And the messenger RNA that is produced at the end of the messenger RNA specifically that is produced by the end of transcription, the process of transcription is known as messenger RNA. So messenger RNA is also known as a transcript. Now notice here again that synthesis of messenger RNA goes from the 5' end to the 3' end. The DNA template is read from 3' to 5'. Notice as well that whenever instead of a T you have a U in the RNA, and that's it. Okay. So the enzyme that is necessary for RNA synthesis or transcription is known as RNA polymerase, and of course there are different types of RNA polymerases. Now what these polymerases do is that they catalyze the formation of phosphodiester bonds between any nucleotides, and the substrates are nucleoside triphosphates, that is ATP, CTP, UTP, and GTP. Now, remember what the substrates are for DNA polymerases. Just like DNA polymerases synthesizing DNA, energy is needed, and energy comes from the substrate themselves. So whenever the two phosphate groups, that is the pyrophosphate group, is removed, you have release of energy, and this energy is the source, is what's necessary for forming the phosphodiester bond between the two nucleotides. Okay. Now there are differences between DNA replication and transcription. One of these differences is that the RNA strand does not remain hydrogen bonded to the DNA template. So you remember that when you synthesize the DNA strand from its DNA template, both of them remain hydrogen bonded. Whereas with the RNA strand, the RNA is synthesized, but it does not remain hydrogen bonded, it gets released. Another difference is that the RNA polymerase reads the A in DNA and it inserts U in the RNA. Okay, complementary to the U instead of adding a T like in the DNA polymerase. The other thing is that the RNA molecules are much shorter than DNA molecules, so DNA is large, huge, it's a whole genome, whereas for the transcription and RNA molecules that are produced from DNA regions, usually we're talking about a small region of DNA that is read to synthesize the RNA molecule. Now, another thing is that unlike DNA, RNA does not store genetic information, and that is important because if there are mistakes in the RNA, it doesn't matter. Okay, we can degrade it and discard it and use something else and make another RNA. But for DNA, we cannot do that. It has to be accurate. Now, there are differences between DNA polymerases and RNA polymerases in terms of substrates, in terms of initiating synthesis of DNA or RNA. For example, DNA polymerases need primers, RNA polymerases do not need primers. They can start synthesis of RNA de novo, that is from scratch. Now, in terms of accuracy, RNA polymerases do little mistakes. They are accurate, but they can still make mistakes. Now, our DNA polymerases are very, very accurate because of proofreading mechanisms. RNA polymerases do have proofreading mechanisms, but they are they have lower accuracy than DNA polymerases. A very important point to mention here is that so this picture shows the binding of the RNA polymerase. So this is the RNA polymerase in blue. So the RNA polymerase binds to the DNA template. It uses one of the strands to synthesize the RNA molecule. Now, as it moves, and it moves in this direction right here, so it's going to the right, as it moves, it starts forming the phosphodiester bonds between nucleotides. So again, the messenger RNA is complementary to the DNA. Now, notice that the newly synthesized nucleotides um, are bound to the DNA template itself. Okay, so this binding, this hydrogen bonding between DNA and RNA is necessary for stabilizing the whole complex. Okay, for stabilizing the interaction, the binding of the RNA polymerase to the DNA and to the messenger RNA. So as it moves forward, you have the older nucleotides released from the DNA template, and you have the new ones binding, or hydrogen bind, the hydrogen bind to the DNA template. Okay. Now notice the bubble right here. So as the RNA polymerase moves forward, you have the DNA, the two strands of DNA separating from each other. Now, during RNA synthesis, during transcription, you have the formation of structures known as polysomes. Again, poly means multiple, zomes like objects or bodies. And so what you have in here is synthesis of RNA from two genes. You have transcription of two genes. That's the one gene, and this is the second gene right here. Okay, and what happens is that you have synthesis of RNA, multiple RNA molecules from the same genes at the same time. Okay, so you have one RNA polymerase binding to the beginning of the gene, and it moves forward, synthesizing the RNA, or it moves forward, it goes this way. So it moves forward, synthesizing the messenger RNA. Now, after it passes through, you have binding of another RNA polymerase. So you have the first one binding and it moves forward, and then you have another one binding and it moves along with it, and you have then a third one binding and it synthesizes RNA. So right here you have multiple polymerases, RNA polymerases binding to that gene, synthesizing the RNA molecules. Okay, so the question is, where is the beginning of this gene? Is it point A or point B? Okay, so look at the length of the RNA molecules. Here the RNA molecules are shorter and they're getting longer and longer and longer and longer. Okay, so this means that this is the beginning of the gene, and RNA polymerases, multiple RNA polymerases moving together sequentially, one after the other, synthesizing the RNA molecules from that gene. Same thing with this gene right here. This is the beginning of the gene. This is the end of the gene. Okay, so you have the RNA molecules getting longer and longer and longer as RNA polymerases move forward, move along the DNA template or the genes. Now here we have many genes right here. So right here you have a gene, here's another one, that's a third one, this is number 4, this is number 5, and so on. So again, here we have a gene right here. This is the beginning of the gene, and the RNA molecules are extending to the outside of the RNA polymerases, and here you have the end of the gene, of that particular gene. Now this thread right here is the DNA itself, and these that are extending outward are the RNA molecules. So this is quite efficient. So you can produce a lot of messenger RNA molecules from all at the same time from the same gene. So they're moving one after the other, and that of course saves time, a lot of time for organisms and cells. Okay, so let's talk about transcription in prokaryotes. So prokaryotic genes can be monocistronic or polycistronic. Okay, so some genes are monocistronic, other genes are polycistronic. So what do we mean by that? So basically what happens here is that for monocistronic genes, basically you have one gene, you have one messenger RNA, and this from one messenger RNA you can have the production of one polypeptide. That's monocistronic. Now, there are other genes that are polycistronic. What do we mean by that? It means that here we have a DNA, you have RNA molecules, a messenger RNA produced from this particular genetic unit or gene. So you have one messenger RNA, okay? Except that this region of the messenger RNA would produce one polypeptide. This region would produce a different polypeptide, a different protein with a totally different function. Here you have a third region right here on the same messenger RNA producing a third and different polypeptide and eventually protein. Okay, so this is polycistronic. So basically you have a genetic unit which is known as a cistron, and this genetic unit produces one messenger RNA, and this one messenger RNA would produce different polypeptides and different proteins with totally different functions. Okay, so this is sort of like a quite efficient system, because these proteins that are produced from the same cistron, the same genetic unit, from the same messenger RNA, these proteins participate in the same function. For example, we'll talk about this later on in terms of metabolism of lactose, synthesis of tryptophan. Tryptophan is an amino acid, lactose is a sugar. So all the proteins that are necessary for synthesis of tryptophan, they're all produced from the same messenger RNA. All of the proteins that are necessary for the metabolism of lactose, they are produced from the same messenger RNA and from the same cistron, the same genetic unit. So this is an efficient system. And that's what we mean by polycistronic. Again, one messenger RNA produced from one cistron, and this one messenger RNA can produce different polypeptides from different regions. Okay, so pay attention to that, because there are RNA variants in eukaryotes as well. Okay. Now, this, by the way, this cluster of genes that exist within the same cistron, within the same transcriptional unit, this one right here, it's known as an operon. Okay, so operons are polycistronic, meaning that they are transcribed as one unit, one messenger RNA, and this one messenger RNA can produce different polypeptides from different regions of the messenger RNA, and these proteins, these polypeptides, are then form proteins, and these proteins participate in the same pathway. Now, how about the RNA polymerase? In E. coli, there is only one RNA polymerase, and this RNA polymerase is made of different subunits, and these subunits are alpha, beta, beta prime, omega. Okay, and these subunits are necessary for they make up the core enzyme, the enzyme that is necessary for transcription. So the core polymerase is capable of catalyzing polymerization of NTPs, that is, N means anything, any nucleotide, any ribonucleoside triphosphates into RNA. Now, there's also another subunit, and this subunit is quite important as well for the function of the RNA polymerase, but it's not necessary for the formation of the RNA molecule, and it's known as the sigma subunit. So the sigma subunit is not necessary for the basic catalytic activity of the enzyme for the RNA polymerase, for RNA synthesis. So how does the RNA polymerase know where to start transcription at? the RNA polymerase binds to a region known as the promoter. So the promoter is the RNA polymerase binding site. And this region exists upstream of the transcription start site. So what is a transcription start site? It includes the first nucleotide that is read by the RNA polymerase. And it is also the first nucleotide of the RNA molecule. And it's known as the +1 site. So the RNA polymerase reads the first nucleotide, and it's known as the +1 site, and it adds a ribonucleoside it uses ribonucleoside triphosphate as a substrate to synthesize or to attach to it the second nucleotide, which is the +2, forming a phosphodiester bond between the two. Okay, and of course there's energy that is produced that is necessary for the formation of the phosphodiester bond. So the second nucleotide is basically becomes a ribonucleoside monophosphate. Then you have +3, then you have +4, +5, +6, etc., and it keeps on going on in this direction. Okay, so the RNA polymerase binds to the promoter, and the promoter region is located upstream of the transcription start site. So it's like a stream, okay? So it's water that is going downstream. So the upstream is up right here, okay, on top. This is so that's how we image promoters and genes. So promoters exist upstream of transcription start sites. Now, or before transcription start site, in other words. So within promoters, there are two regions that are important, and these regions are composed of consensus sequences. So a consensus sequence is basically a sequence that exists in different bacterial species. And that means that this sequence is important. It has functional significance. Okay, so there are two consensus sequences within the promoter, and these sequences are known as the -10 region and the -35 region. So why are they known as -10 and -35? Because the -10 it includes nucleotide number 10 right before the transcription start site or upstream of the transcription start site. Okay, now the -35 region comes 35 nucleotide, or it includes nucleotide number 35 that comes right before the transcription start site or upstream of the transcription start site. Now we can also say that this region, the -35 consensus sequence, comes upstream of the -10 region. Okay, so these regions are actually whereby the RNA polymerase bind to exactly. So this whole region is known as the promoter, but interaction between the promoter, between the RNA polymerase with DNA exists at these two sites right here. Okay, so these regions are important. If you change any of the nucleotides within the -10 or -35 regions, that would compromise the interaction between the RNA polymerase with the promoter, and that would affect the efficiency of transcription. So I said that the sigma subunit is not important for the catalytic, for the function of the enzyme, but it does have importance though. And the importance of the sigma subunit is that if it's removed, if it does not exist, the RNA polymerase would have low affinity to the promoter, and it would bind to the promoter non-specifically. So what does the sigma subunit do? What it does is that it guides the RNA polymerase to the -35 and -10 regions. So it strengthens, it enforces the interaction between the RNA polymerase to the promoter. Okay. So here's what happens. The sigma subunit binds to the RNA polymerase. Okay. And then it guides it to the promoter region, forming what is known as the closed promoter complex. So now you have interaction between the RNA polymerase with the promoter region, the -10 and -35 regions. The RNA polymerase itself pulls apart the two strands. It separates the two strands from each other, forming the open promoter complex. Now you have this bubble right here. Once you have this bubble, the RNA polymerase can initiate RNA synthesis. So it reads the +1 site and it adds the first nucleotide, and then it reads the second, it adds the other nucleotides, forming phosphodiester bond between the two. Then it adds the third, the fourth, the fifth, and so on, and it keeps on moving forward. Now, after reading about 10 nucleotides, the sigma subunit is released, because now the RNA polymerase does not need it anymore. It's guided, it's already guided. So what does happen to the sigma subunit then? Does it get degraded? No, it does not. It binds to another RNA polymerase and it guides another RNA polymerase to the promoter region. So as this moves forward, you have the second one binding to the promoter region, and you have another round of transcription. So you have two polymerases doing RNA synthesis at the same time from the same gene. Now, once the second RNA polymerase passes through 10 nucleotides or so, the sigma subunit is released and it binds to a third RNA polymerase and so on. So it guides it to the promoter, okay, and so on. So you have like 3 RNA molecules synthesized at the same time from the same gene. You have the second stage of transcription, which is elongation. Now, so for transcription and even for replication, by the way, and even for translation, we can divide the processes into 3 phases. We have initiation, we have elongation, and we have termination. Okay, so in the second stage right here, elongation, it's the RNA polymerase that does a lot of things at the same time. So what it does is that it unwinds the template DNA, it separates the DNA strands from each other, so it functions like what? What is the enzyme that separates the two strands of DNA from each other? It's known as a helicase. So you have the RNA polymerase putting apart the two strands from each other. It synthesizes the RNA, it elongates the RNA. Notice that you have this interaction between the newly synthesized messenger RNA or nucleotides with the DNA template. Okay, and this stabilizes the interaction between the 3 components, that is the RNA polymerase with the DNA and the messenger RNA. And the RNA polymerase does the third thing, which is rewinding the DNA right behind it. So it separates the 2 strands from each other, it synthesizes RNA, and it rewinds the DNA. It closes the 2 strands from each other. Now you can have another one right here, and another RNA polymerase right behind it, but you will have double-stranded DNA between them. So it's not like the DNA is fully separated. And then you have the third stage of transcription, which is termination. That is, the RNA polymerase stops RNA synthesis. So the question is, how does the RNA polymerase know that it should stop transcription right at this point? Now it knows that it should terminate synthesis of RNA from a termination sequence, and it is a consensus sequence again, because you can find the same sequence at the end of different genes, and you can find it in different species as well, bacterial species. And this region is a consensus sequence. Okay, so now this sequence is GC rich. Okay, and it is followed by a stretch of U's. If you look at the gene itself, the sequence is basically A. Okay, again, GC rich followed by A. Now, so what happens with the GC rich region is that it forms what is known as a stem loop structure. Okay, so you have this stem loop structure that is formed. The RNA forms this stem loop structure. So you can imagine the stability of the stem loop structure because it is rich with G's and C's. So this structure is quite strong. And this is followed by the stretch of U's. Okay, now these U's have two double bonds with the A's in the DNA template. So you can imagine that the interaction between the messenger RNA with the DNA with the RNA polymerase is quite weak. It's not as strong as the GC-rich region. So let's put it all together. What is really important about this interaction? So I said right here you have the formation of the stem loop structure, and this stem loop structure is quite stable. It's very strong because of the G's and C's. And then you have U's, and these U's interact with the A's, meaning that relatively this interaction is weak. So what happens is the following. I keep I imagine it as like an Indian movie or a Hollywood movie. So basically what you have is the following. You have the hero running in a green field or something. So that's my imagination. Okay, so you can imagine that the hero is running from a monster and in a green field or something, and this hero is looking backward, and he doesn't see anything, doesn't see any monster or anything, and all of a sudden you have this monster coming out of the ground, and it's going like this. Woo. Okay, so as it's going out of the ground shakes. Now the hero reaches a region that is slippery. Okay, this is where the U's are, and the monster comes out, and the hero reaches this slippery region right here. So he falls around. Okay, he falls down because he can, it's slippery, it's the ground is shaking, and the interaction between the hero with the ground is weak. Okay, so the interaction between the U and the A is weak relatively, right? And because you have this stem loop structure coming upward, and the interaction becomes weak at the U region, now the messenger RNA dissociates from the DNA. And once you have dissociation of the messenger RNA from the DNA, the whole RNA polymerase dissociates as well, and that's the end of transcription.